When we sequence a genome, what do we actually get? At first, we only get a long string of letters, A, T, C, and G. That sequence is valuable, but by itself, it does not tell us what the genes do. This is where functional annotation becomes important. Functional annotation is the process of assigning biological meaning to raw sequence data. It helps us move from sequence to function. In simple words, it helps us answer questions like, what gene is this? What protein does it encode? What does that protein do inside the cell? This process usually combines two approaches. One is experimental evidence, which is the most reliable. The other is computational prediction, which allows us to analyze very large datasets efficiently. So if sequencing gives us the alphabet of biology, functional annotation helps us read the language. Now this raises an important question. Why is sequence alone not enough? Imagine walking into a huge library where none of the books have titles, labels, or sections. All the information is there, but nothing is organized. That is what a genome looks like without annotation. Functional annotation gives structure and meaning to genomic data. It allows researchers to interpret genes and proteins in a biologically useful way. This becomes especially important in omics studies. For example, if you perform an expression analysis and identify 500 upregulated genes, that list alone does not explain the biology. But once those genes are annotated, you can begin to see pathways, cellular processes, and disease mechanisms. That is why annotation is foundational for genomics, transcriptomics, disease research, and drug discovery. If you look at the concept on the slide, the key message is simple. Annotation transforms raw data into interpretable biology. To do that well, we need to annotate several different layers of information. Functional annotation is not just one label added to a gene. It is multi-layered. We may want to know the function of a protein, the domain it contains, the cellular location where it acts, and the interaction partners it works with. In other words, we are not just asking what the molecule is. We are also asking how it works, where it works, and with whom it works. For example, a protein located in the nucleus may be involved in gene regulation, while a membrane protein may participate in transport or signaling. Similarly, a protein may function alone, or it may act as part of a larger complex. If you look at the slide, think of annotation as building a biological profile from several connected layers. But for this kind of system to work globally, scientists need a shared language. That is where gene ontology comes in. One of the biggest challenges in biology is consistency. Different scientists may describe the same biological process using slightly different words. That creates problems for databases, computational analysis, and cross-study comparison. The gene ontology, or GO, solves this problem by providing a standardized vocabulary for describing gene and protein function. This means researchers around the world can describe biological functions in a consistent way. GO is also structured hierarchically. It starts with broad concepts and moves toward more specific terms. That hierarchical structure is very useful for computational analysis because it allows software tools to group genes into meaningful functional categories. If you look at the slide, GO acts almost like a biological dictionary shared across genomics and bioinformatics. GO itself is divided into three major categories. Every annotated gene can be described from three major perspectives. The first is biological process. This describes the larger process the gene contributes to, such as cell cycle, apoptosis, or glucose metabolism. The second is molecular function. This describes the specific biochemical activity of the molecule, such as ATP binding, enzyme activity, or DNA binding. The third is cellular component. This tells us where the molecule acts, such as the nucleus, mitochondrion, plasma membrane, or ribosome. So one category tells us what broader process is happening, one tells us what the molecule is doing, and one tells us where it is working. If you look at the slide, each gene is telling a three-part biological story. Now the next step is understanding how these GO terms are actually assigned. The GO annotation process usually begins with a sequence, a protein, or a list of genes. The first step is to search existing databases for evidence. That evidence may come from sequence similarity, domain prediction, experimental studies, or curated protein records. Once that evidence is found, it is mapped to relevant GO terms. After many genes are annotated, researchers often perform enrichment analysis. This helps identify which biological functions are overrepresented in a dataset. For example, if many genes in your list are linked to inflammation or cell division, enrichment analysis will highlight that pattern. If you look at the workflow on the slide, the process moves from raw sequence to evidence, then from evidence to annotation, and finally from annotation to biological interpretation. A workflow like this depends on strong reference databases. One of the most important of these is UniProt. If you work with proteins, UniProt is one of the most important databases you will use. It is one of the main global resources for protein sequence and functional information. A UniProt entry usually brings together many layers of knowledge in one place. It can include the protein sequence, name, function, domains, subcellular location, post-translational modifications, interactions, and links to other databases. This makes UniProt a central hub for functional annotation. If you look at the slide, think of UniProt as a detailed biological identity card for proteins. But it is also important to understand that not all UniProt entries are curated in the same way. UniProt contains two major types of entries, and the difference between them is very important. The first is Swiss-Prot. These entries are manually curated by experts. They are highly reliable and usually supported by literature and detailed review. The second is TrEMBL. These entries are generated through automated annotation pipelines. They cover a much larger number of proteins, but the annotations may be less precise than expert-reviewed records. So in simple terms, Swiss-Prot offers higher confidence, while TrEMBL offers larger coverage. If you look at the comparison on the slide, this reflects a common trade-off in biology: depth versus scale. Now, while UniProt focuses strongly on proteins, genome browsers help us see genes in their genomic context. The Ensembl genome browser allows us to view genes directly within the genome. This is important because function is often linked not only to sequence, but also to genomic structure and position. Ensembl shows features such as exons, introns, transcripts, regulatory regions, and genomic variants. It also supports comparative genomics, allowing researchers to compare genes across species. For example, you can examine whether a human gene has a similar structure in mouse or zebrafish. If you look at the slide visual, think of Ensembl as a genomic map that places genes in their physical neighborhood. Another important resource that integrates annotation in a centralized way is NCBI Gene. NCBI Gene acts as a central entry point for gene-related information. It links gene identity with genomic location, functional annotation, literature, pathways, and sometimes protein structure. One major advantage of NCBI Gene is its strong integration with PubMed and other NCBI resources. This allows researchers to move quickly from a gene record to relevant scientific papers and supporting evidence. So instead of searching across many disconnected resources, NCBI Gene provides a more unified view. If you look at the slide, the important message is that NCBI Gene connects sequence information with biological evidence and published knowledge. Now, to understand protein function more deeply, we need to look inside the protein itself. Protein function is often built from smaller conserved units. Two of the most important are motifs and domains. A motif is a short conserved sequence pattern. It is usually associated with a specific biochemical role, such as binding a metal ion or recognizing DNA. A domain is larger and more structurally independent. Domains often fold into stable units and may carry a complete functional role on their own. You can think of motifs as small signatures, while domains are larger functional modules. This is extremely useful in annotation. If an unknown protein contains a known catalytic or binding domain, we can often make a strong prediction about its likely function. If you look at the slide, the key message is that protein function is modular, and evolution often reuses successful structural units. To identify these units, specialized databases are used. One of the early databases used for functional motif detection is PROSITE. PROSITE identifies conserved sequence patterns using defined rules or pattern signatures. These patterns are especially useful for recognizing short functional motifs in proteins. For example, if a protein contains a sequence pattern known to bind ATP or zinc, PROSITE can help detect that. This was one of the first major systems linking recurring sequence patterns with biological function. If you look at the slide, PROSITE represents an early but important step in pattern-based annotation. However, not all proteins can be captured using simple pattern matching. More complex proteins require more flexible models. SMART is a database and analysis tool designed to identify signaling and regulatory domains in proteins. Unlike simple sequence pattern matching, SMART uses more advanced probabilistic models, including hidden Markov models. That makes it better at identifying domains, even when the sequence is somewhat variable. SMART is especially useful for proteins involved in signaling, extracellular communication, and membrane-associated functions. So if a protein participates in receptor signaling or intracellular communication, SMART can often detect the relevant functional modules. If you look at the slide, think of SMART as a tool that helps decode the signaling language of the cell. But researchers often do not want to search every domain database one by one. That is why integrated resources were developed. InterPro is a very important integrative resource in functional annotation. Instead of relying on only one domain or motif database, InterPro brings together information from multiple sources, including PROSITE, SMART, PFAM, and others. This creates a more unified and reliable view of protein domains and families. If several databases independently support the same domain assignment, confidence in that annotation becomes much stronger. InterPro also links proteins to families, functional sites, and GO terms, so it serves as both an integration platform and an annotation engine. If you look at the slide, InterPro acts like a central interpreter that combines multiple lines of evidence into one functional conclusion. Once we know the parts of a protein, we next want to know how those proteins work together in larger systems. Proteins do not work in isolation. Most biological functions emerge from interactions between many genes, proteins, metabolites, and signaling molecules. That is why functional annotation must go beyond individual molecules and move toward pathways and networks. A single protein may be part of a signaling cascade, a metabolic route, or a transcriptional regulatory system. By studying these pathways, we begin to understand how cellular behavior emerges from many connected components. If you look at the slide, the shift is from single building blocks to coordinated biological systems. Two of the most widely used pathway resources are KEGG and Reactome. KEGG, or the Kyoto Encyclopedia of Genes and Genomes, is one of the most widely used pathway databases in biology. It provides visual maps of metabolic pathways, signaling pathways, and disease-related biological systems. This makes KEGG extremely useful for enrichment analysis. For example, after identifying differentially expressed genes, researchers can test whether those genes are enriched in pathways such as PI3K-AKT signaling, apoptosis, or insulin signaling. If you look at the slide, KEGG is essentially giving us a functional map of how molecules connect inside the cell. It helps move us from a gene list to pathway-level interpretation. For even more detailed reaction-level information, Reactome is especially valuable. Reactome is another major pathway database, but it often provides more detailed mechanistic information than pathway maps alone. It is carefully curated and focuses on the specific molecular events that occur during biological processes. Instead of only saying that two proteins are connected, Reactome may describe the actual reaction, binding event, transport step, or modification involved. This makes it extremely useful when researchers want a deeper mechanistic understanding of cellular function. If you look at the slide, Reactome can be seen as a highly detailed reaction library for biology. At this point, it helps to step back and look at the overall annotation workflow as a full pipeline. Functional annotation usually follows a connected workflow. We may begin with raw DNA or protein sequences. Next, we identify motifs, domains, or structural features using tools such as InterPro or SMART. Then we assign GO terms and link proteins to broader biological categories. After that, pathway databases such as KEGG and Reactome help place those molecules into biological systems. So the entire process moves from raw sequence to structure, from structure to function, and from function to systems-level interpretation. If you look at the slide, the pipeline is showing how biological meaning is gradually built layer by layer. That brings us to the practical question: where is all of this used? Functional annotation has major real-world applications. One of the most important is biomarker discovery. If a gene or protein is consistently associated with a disease, annotation helps us understand what role it plays and whether it may serve as a diagnostic marker. Another key application is drug target discovery. If annotation reveals a critical catalytic domain, signaling receptor, or disease-associated pathway, that information can guide therapeutic development. This is especially important in cancer biology, infectious disease research, and precision medicine. If you look at the slide, the key message is that annotation turns data into actionable biological insight. To conclude, we can think of annotation as building a hierarchy of meaning. Functional annotation works by building biological understanding step by step. At the most basic level, we begin with motifs and domains. These are the structural and functional building blocks. Next, we move to proteins and GO terms, where molecules are assigned functions, locations, and biological roles. Finally, we move upward to pathways and networks, where we understand how many molecules work together to produce cellular behavior. So functional annotation is not just about naming genes. It is about transforming raw sequence data into an interpretable map of life. If you look at the final slide, the hierarchy moves from small features to full biological systems. That is the real power of functional annotation. Thank you.